Okay, so here's a little run through and review of meiosis. Alright? First thing I'm going to do is I'm going to draw up here a chromosome. And remember, we have to imagine that this chromosome is one long single strand of DNA that's tightly wrapped around these proteins, and then these long strings of proteins that the DNA is wrapped around, these histones, are tightly wrapped and coiled around with one another, forming this big structure that kind of resembles something like this. Okay? And inside your body, you've got 46 of these. Okay? You've got 46 of these, and there's 23 pair. So there's another one that looks very similar to this, and probably also encodes for some of the same information. Okay? So again, this is one piece of coiled up, very tightly coiled up DNA. Much more tightly coiled up than I'm showing right here. Okay? So on this, alright, we can break this up, and you'll see it represented this way in a lot of what we're looking at over the next couple weeks. We say that this little area right here, this purple area, might encode for hair color. Okay? Like it's the gene that encodes for hair color. As is this purple area on this chromosome. Okay? So this could be like the first and second chromosome. So numbers 1 and 2 of 46. Alright? Or it could be considered the first pair. Alright? The first pair of 23. Okay? So these guys look very similar to each other. We call them homologous chromosomes. This would be a homologous chromosome. And if we were to redraw this guy, as we see it when we're looking at meiosis, it would look something a little more like this. Now these guys get really close to one another. They don't cross one another during meiosis and mitosis. They get really close to one another. There's these proteins that really do work and kind of connect these guys. And that area, again, of connection is called the centromere. All right? There's some other vocab terms, kinetochore, synapses, but we don't really need to look at those. We're just talking about this area we know as the centromere, being where the two homologous chromosomes, the two chromosomes that are like each other, come together. Okay? We also talked about how DNA replication occurs. So at the very beginning of meiosis, DNA replication has happened. So we not only have 46 individual chromosomes, or 23 pair, we've actually got 92 individual chromosomes, and we've got 46 pair. Alright, we've got 46 pair. And we know that the homologous chromosomes find their other homologous buddy, and they make a homologous pair. So it looks something a little more like this. Okay? And those guys line up on the equator. Now crossing over occurs. Okay? Crossing over occurs in prophase 1 of meiosis. Alright? So these homologous chromosomes, this HC and this HC, come together to make a homologous pair, an HP. Alright? And reciprocal or corresponding areas of the homologous chromosomes are then flip-flopped. So this section over here may go over here, this section over here may go over here. It's called crossing over. Okay? This section of genetic material, this gene right here and this gene right here, they may switch with one another. Okay? Between homologous pairs. Alright? Big stage there to promote genetic diversity and genetic variation for the sex cells that meiosis is going to create. Okay? So meiosis is underway. We just took care of prophase 1. The examples I'm further going to draw up here, I'm not going to draw all 46 pairs at the very beginning. Okay? I'm not going to draw and include all 92 individual chromosomes. But we still have to imagine, a lot of times when we're talking meiosis, we're talking stuff that is dealing with humans. We're talking the human race here. Okay? But what I'm going to draw up here from now on will not represent that. I'm not going to put that many chromosomes up there. Alright? But remember, 2N means you have 2 sets of the DNA. And for human beings, it is normal for us, it is typical that each one of our cells has 2 sets of the DNA. 2N being diploid, N being haploid. And our sex cells are haploid. They are N. Okay? They're just one set. Alright? So meiosis, right here at the beginning, DNA replication has occurred, prophase 1. is all about that crossing over, and we just showed that. Okay, and you also have to think at the very beginning here, alright? At the very beginning here, and I'm just going to use 2 sets here. Alrighty? We are actually 4 in, believe it or not. Just for a brief minute, and that's because we've gone through that DNA replication. We've actually got 4 sets of DNA in here right now. Okay? Alright, 4 sets of DNA. But again, we're not going to diploid cells like we would be in mitosis in the regular cell cycle. We're going to haploid cells. Alrighty? So prophase 1 occurs. This is actually be metaphase 1 because I've got these guys lined up on the equator. Alright? So metaphase 1, these guys are lined up on the equator. This homologous chromosome from this homologous pair will go to this pole, this one to this pole. Okay? This being metaphase 1. And then anaphase 1 would be represented by the arrows, okay, the actual going. I'm not going to show that, but it's the actual going to the poles. And then telophase 1, we would start to split this cell in half, okay, and new nuclei form around the homologous chromosomes. That's important to mention, the homologous chromosomes, okay, because they're still in that state. They may unravel, but they would just coil back up into form homologous chromosomes once again. Okay? And this would be telophase 1 right here. Alright? And we'll say cytokinesis does happen and splits that guy in half. So we go back up here, and now these guys have become 2 cells, so we've got 2 cells. Prophase 2 will take place. Prophase 2, there is no crossing over. And do not forget that between these stages of meiosis, DNA is not replicated. DNA is not replicated. So we've actually gone from 4 in at the very beginning of meiosis to now having 2 cells that are each diploid. Okay? 2 cells that are each diploid. So prophase 2, the chromosomes, if they have uncoiled, they do coil back up, condense back up to form chromosomes. Those chromosomes find their homologous chromosome, and then in metaphase 2, which I'm going to draw right here, okay, metaphase 2, those homologous chromosomes line back up on the equator. And again, the cell of the equator could be in a couple different places. I'm going to draw it horizontally with this cell, I'm going to draw it vertically with this cell. Okay? So anaphase 2 is similar to anaphase out of mitosis, okay? This homologous chromosome right here is now going to split at the centromere. It's going to split at the centromere, and that individual chromosome, or chromatid, is now going to travel to the pole. So this guy ends up traveling here, and we can't forget the spindle fibers. I haven't mentioned those one time yet, but we know it's the spindle fibers that pull those individual chromosomes to their poles. And we end up getting telophase looking something like this, where now we've got nuclei forming around these individual chromosomes. Okay? And each one of these new haploid sex cells is going to look like this. With just 2 individual chromosomes inside the nucleus of each one of those cells. Alright? It's important to follow the vocab. Main thing I wanted to do was to put out a video of me going through meiosis for you. Everything I've seen so far looks pretty good from you guys. I just really wanted to push the homologous pairs to homologous chromosomes to individual chromosomes. And we do not reline back up on the original equator from meiosis 1. I saw that a lot in the videos that you students have made so far. So I wanted to clarify that. If you have any questions, by all means, come and ask me. I guess I didn't mention independent assortment. That would just be how these guys right here, the homologous chromosomes in metaphase 2 and the homologous pairs in metaphase 1, just randomly distribute themselves across the equator. There's no rhyme or reason to how they line up on the equator. They just line up on the equator how they see fit. Okay? That's meiosis. Got questions? come and ask me. Later.